Hello, I am François Auréat. I am the NanoSIMS product manager at Kamika. And in this webinar, we will illustrate the main NanoSIMS capability described in the part 1 of this webinar. In the coming slides, we'll go through sensitivity with high lateral resolution, elemental mapping from light elements, atmospheric elements, up to very heavy elements, and we will see the isotopic ratio mapping in material science and life science. For this first example, we will use a thick piece of silicon nitride, which is electrical insulator, and we were asked to map some dots, unknown dots, on inside this material. So this analysis was tricky before with other techniques. For TEM, sample thinning was not so easy, and for light elements, sensitivity was not very good. Auger was not usable because of sample charging. So we see here an image of carbon, boron, nitrogen, and silicon. Note that boron and nitrogen are imaged through small molecular ions. This field of view is 6 by 6 micron. The spot size is 100 nanometer, and the image was recorded in 22 minutes. What we can see from this, all these data are collected at high mass resolving power. So this is the first point, nearly no mass interference at this low mass range. If we look at the highest intensity in the 3 images of carbon, nitrogen, silicon, we see that there are more than 10,000 counts per pixel, per single pixel. So what does it mean? Let's assume that it's 100% pure carbon or pure nitrogen or pure silicon. That is not the case, but let's assume this. It means that we, if we divide 100% by 10,000, we are in the 100 ppm range for 1 pixel of 100 nanometer size. So this is the exact interest of the NanoSIMS, the sensitivity together with a small size. So now it's, if you add maybe only 10 by 10 pixel, so 0.3 micron size, you enter in the ppm range detection limit in imaging mode in 22 minutes. So that's from one single example, insulating sample. You can travel on the surface, no need for sample prep. Sample just must be flat, short analysis time, and good detection sensitivity. One of the unique points of SIMS is its capability to analyze hydrogen and deuterium. So I will illustrate this using a scientific paper by Claude Lochan, working with the NanoSIMS at Harvard Medical School. And here you see an endothelial cell imaged through the ion Cn-, imaging proteins. And if you repeat the measurement, you will progressively penetrate inside the material. And this is what you see on the image on the right. And this is an image after some sputtering. You have penetrated inside the cell and you superimpose 3 different maps recorded in parallel: carbon, nitrogen, phosphorus. And if you select hydrogen, you will be able to record also hydrogen mapping from any materials. Here it's a cell, but it can be metal or any other material. And this is why we have in the NanoSIMS ultra-high vacuum chamber to avoid any contamination from the residual gas. We can continue to increase the mass, switching to lithium. Lithium is an element that is not so easy to analyze with many other techniques. Here, we use an article produced by Mr. Crum at Pacific Northwest National Lab in Richland in USA, and they were working on glass ceramics for storing nuclear wastes. So what we see in this study are images of elements, light elements, lithium, boron, aluminum, silicon, and the second row of images, a zoom of the upper row. The interesting point is that you can really, without any problem, image lithium with the NanoSIMS. That's one point. The sensitivity is extremely good because lithium ionizes very well. And you can also detect other elements, and we see in this example, in this class, boron is imaged with BO minus, because here it was imaged with cesium primary, and aluminium as aluminium O minus, because there is a lot of oxygen in the oxide, and so, but it could be also analyzed with oxygen primary beam instead of cesium, and in such case we would use lithium plus, boron plus, aluminium plus. So good sensitivity for light elements, including lithium. What we can add also is that as it's an insulator, it's possible to analyze such sample in the NanoSIMS using an electron float gun. That is one option of the instrument. And generally, we use also metal coating, a thin layer of metal coating, platinum, for example, to help in charge dissipation. In order to illustrate the large range of accessible elements, I took this scientific paper published in Science around plutonium. And here, the researchers focused on analyzing colloids found in groundwater at a few kilometers around a nuclear plant in Russia. And what they used for technique, analytical technique, they first used TEM to visualize the colloid, to identify the colloid, then EDS to identify the elements, and then the major elements, and then the NanoSIMS was used, and you see here a map showing aluminium, manganese, calcium, iron, and uranium and plutonium imaged through their iron UO minus and plutonium O minus, as they were imaged with cesium plus primary ions. So this example illustrates well the capability of the NanoSIMS to first detect the masses. NanoSIMS can detect ions from mass 1, hydrogen, to mass 400 at 16 keV and higher if you reduce the primary energy. And also, it illustrates the sensitivity of the NanoSIMS to detect heavy masses from small objects in the environment. And here, the conclusion was that phosphorus iron oxide colloids absorb plutonium hydroxides or carbonates along with uranium carbonate. Based on mass spectrometry, the NanoSIMS permits also to use or to map isotopic ratio of one element. So I use this publication from the University of Gothenburg in Sweden, where they are studying the oxidation mechanism of a metallic alloy. In order to study this, they use a dual oxidation step using one phase of oxidation in O18 rich environment and one phase of oxygen-16 rich gas environment. So they have two samples, one for a shorter oxidation duration. So first oxygen-18, second oxygen-16, 168 hours, and a second one, 500 hours of oxidation. This can be studied and was studied by direct depth profiling using an NanoSIMS 5. And you see the depth profile showing the surface enriched with oxygen-16 and the intermediate layer oxidized with oxygen-18, but depth profile doesn't bring much information. Why? This can be explained by looking at the TEM cross-section. You see that the surface and the interfaces are extremely inhomogeneous. So the depth resolution is totally destroyed by this roughness. Instead, one can use a different strategy, a strategy of correlative microscopy, combining TEM, giving a resolution of maybe 1 nanometer, and NanoSIMS, based on the cross-section image. So then one can superimpose oxygen-18 image in green, oxygen-16 image in blue, and from these images, one can draw lines and reconstruct line scan in any direction. And here you can see the different layers with the interdiffusion of the elements, oxygen, oxygen-18 in green, oxygen-16 in blue, and the depth resolution is given here by the cesium primary beam diameter around 50, 100 nanometer. So good depth resolution despite a huge topography. For the second sample oxidized for a longer time, 500 hours, we see the TEM cross-section. And when we look at the oxygen-16, we see clearly a path of oxygen-16 along the grain boundaries. When we superimpose the oxygen-16 in blue and oxygen-18 in green, we clearly see the penetration of the oxygen along the grain boundary. So we can see with this, with the good spatial resolution and the correlative information from TEM, we can have an information of the mechanism of the oxide growth along the grain boundary as here or through the grains. The NanoSIMS is an imaging instrument, but it is also, by definition, a SIMS instrument, so a destructive instrument, so it can be used as a depth profiling instrument. So I show this example of a B implant, boron and phosphorus implant in silicon. It's a standard that we use to illustrate the flexibility of the conditions for depth profiling in the NanoSIMS. So here we superimpose different profiles on the same sample, recorded with cesium plus, BSI minus, or oxygen minus B plus, or oxygen 2 minus B plus, or oxygen plus BO2 minus. So a variety of conditions that can be used depending on the priority of the analysis. So the NanoSIMS requires opposite polarity always between primary and secondary. So it's good to have this flexibility. Depending on the condition, we will promote one aspect of the analysis. So depth resolution will be better with heavy primary ion or molecular primary ions. And speed will be better with heavy primary ion, fast pattern. Useful yield will depend. Mostly, cesium is used for electronegative. Oxygen is used for electropositive secondary ion. The impact energy can be selected on the NanoSIMS, presently between 4 kV and 16 kV. 16 kV for best spatial resolution, 4 kV for better depth resolution. And from the superimposition of the curves, one can see that the detection limit doesn't vary much. In this example, around 1 E16 atom per cubic centimeter, so 0.2 ppm per detection limit for both boron and phosphorus. And this, while we see this, we can mention that all the NanoSIMS data are recorded at high mass resolution. So we do not suffer from Si30H mass interference on phosphorus because we work at high mass resolution. The NanoSIMS has this specificity to keep good sensitivity even when we work at high mass resolving power. So the key point of these profiles are that they are recorded from a small area, here 15 by 15 micron crater, but it can be reduced down to our maximum, let's say 1 square micron, not much below. In order to illustrate what the NanoSIMS can do in life science, I selected this paper issued from a collaboration between the Museum of Natural History in Paris and the EPFL in Lausanne, both equipped with NanoSIMS, working on coral and the symbiosis between coral and dinoflagellate and those symbionts. So these corals are grown in aquarium, and after preparation, they are embedded in resin and microtome. And on the right side, you can see an optical view of stained coral tissue section. So in order to analyze the interaction between the coral and the dinoflagellate and those symbiont, sections were prepared after incubation or spike of nitrogen-15 labeled ammonium, and then chase with normal seawater. So the idea is to see the response of this living system after a spike of nitrogen. That will be nitrogen-15, so we'll be able to trace it with the NanoSIMS, measuring and following the evolution of the nitrogen-15 over nitrogen-14 ratio in the tissues. So here you see the two sections analyzed and correlated with TM on the left for the best resolution and NanoSIMS isotopic map on the right, showing the evolution of the ratio. Here, the storage or the accumulation of nitrogen, nitrogen from the spike. And this is a good example of correlative microscopy that is used to study living interaction. Then, by extracting samples at various time of the experiment, it is possible to measure dynamics. So in this study, samples were extracted at different time of incubation with the nitrogen 15 minutes, 30 minutes, 45 minutes, 60 minutes, and one can see the storage of the nitrogen 15 on the seawater by the endosymbiont. Then in the chase phase, so the rinsing with seawater, normal seawater, it's possible to see the remobilization, 1 hour, 5 hour, 11 hour, 23, 47, 95 hours of remobilization of nitrogen from the storage into the main tissue of the dinoflagellates. So this strategy is a general strategy in life science of a pulse chase experiment with sampling at various times of the experiment. In environmental microbiology, combining different optical microscopies, confocal, fluorescence, can be extremely powerful. Here we show some results from an article published by the Lawrence Livermore National Lab group in the USA. It's about the identification of a cyanobacterial group in a microbial mat. So the strategy was to incubate this mat with gas of nitrogen 15 and see where it could be fixed in the different cells in the field. So one can use different fluorescence microscopy. The first one is DAPI to localize cells. So cells were localized with DAPI. Then one can go one step further, is to use cardfish. And here one will identify the microbial identity, so through the RNA 16S. And once this is done, one can run the same measurement, same imaging analysis in the NanoSIMS. On the left, you can see a secondary ion, secondary electron image induced by the primary ion, and on the right, you can see the nitrogen 15 over nitrogen 14 ratio, and you can quantify the fixation, so the metabolic activity of single microbiome. And so you have both information. So the identity of the microbes and, in this case, bacteria, and its metabolic activity on a given substrate. So here fixing the gas, nitrogen gas. And one thing that is quite important to mention is that this single cell analysis does not require any cell culture. So even single cells or rare or slowly replicating organisms are accessible through this strategy. Let's conclude this webinar. The NanoSIMS 50L enables trace element and isotopic ratio, high resolution mapping of most elements, from hydrogen, atmospheric species, lithium, up to plutonium and higher, from flat and dehydrated samples. It also, as we have seen, permits this analysis over depth profile information. And this instrument is really used in a very large diversity of applications. Very often, the NanoSIMS is correlated with different microscopy images. the electron microscopy, SEM, TEM, STEM, that gives you maybe nanometer scale spatial resolution. And in life science, it is extremely often correlated with optical and fluorescent microscopy. And FISH is one of the most interesting techniques for microbiology to identify the bacterial species. The NanoSIMS offer unique capabilities where other imaging mass spectrometry instruments lack sensitivity and or mass resolving power. For example, in time-of-flight SIMS, where you miss sensitivity, you miss mass resolving power. Quad SIMS, you miss resolving power. MOLDI, you will not access the low mass range, and you will miss resolution. In some other technique, like magnetic sector SIMS or laser ablation ICP-MS, you will miss really the lateral resolution. Mostly the problem of resolution. With instruments offering higher spatial resolution, like SEM, TEM, combined with EDS or EELS, or field emission Auger, you might miss light element detection or sensitivity. Lithium, nitrogen, boron, carbon, for example. You will miss also with this technique the isotopic ratio capability that offers so many possibilities, new possibilities for researchers in the NanoSIMS. And also, these techniques can suffer from the weight or the limitation of the need of sample prep. The NanoSIMS, you do not need to prepare much the sample, except that it must be flat. Thank you for your attention. So please do not hesitate to watch other webinars or to contact us for more information.